Best regards to everyone. Our project is automatic curation of LTR-retrotransposons libraries from plant genomes through machine learning techniques. It was developed by Mariana Sofía Candamil, Paula Andrea Jaimes, Estiven Valencia, Reinel Tabares-Soto, Simón Orozco-Arias, Romain Guyot, and Gustavo Isaza. We belong to Universidad Autónoma de Manizales and we are part of the Bioinformatics and Artificial Intelligence team. Transposable elements are mobile genetic entities because they have the ability to change from one chromosomal location to another. There are multiple classifications of these elements. However, the order that interests us most are the LTR-retrotransposons because it is the most abundant in plant genomes. The transposition can have a great impact on the expression and regulation of genes. Also could lead to change the structure of the element and even produce nested insertions, which don't represent a reference for future identification and classification studying. Considering the great importance of transposable elements in species, several techniques and methodologies based on bioinformatics have been developed. That is using computational tools in order to study these elements in depth. But these types of methods require a huge time of execution. For this, the objective of this project is to design a method based on machine learning to cure DNA sequences in plants. That is to identify the LTR retrotransposons that are of low quality and that have mutated over time and separate them from those that are of good quality and can represent a reference for future identification and classification studies. For the methods, we first create a plant LTR sequence dataset. For this, we use ImpactorDB dataset, which has the intact or good quality sequence of LTR retrotransposons of 195 plant species. And likewise, using Impactor software, low quality sequences were identified using several filters including size and the presence of domains. For the second stage, we did experiments using machine learning models, taking into account that the models that are on the screen. Then we did experiments using fully connected neural network, obtaining the architecture that consists of 3 layers, each one conformed by 200 neurons, including other parameters like batch normalization and ReLU activation function. And finally, we did generalization test, using 4 genomes to test the performance of the model. We obtained that for conventional machine learning models, the best F1 score was for multi-layer perceptron, with a value of 90.39%. In order to increase that percentage, we proved the fully connected neural network in which we obtain a F1 score of 91.18% in the test data. Thanks to the training curve, we can notice that the generalization works correctly. Also, to rectify that information, we execute the test with several genomes. Considering that results, we obtained that the best results were for Oryza granulata, in which we highlight the percentage of F1 score of 93.6% and an accuracy of 92.9%. Also, in the confusion matrix, we can appreciate that the main diagonal gives higher values than the secondary diagonal, also demonstrating the good generalization of the model. Finally, We can say that we develop a tool to filter LTR-retrotransposons libraries, reducing significantly the execution time, which allows to analyze more genomic data of species and thus to understand the evolution, adaptability to environmental conditions, and contribute for future studies related to identification and classification of these elements.